Hello and welcome to the Investigative Studies Program Fall Symposium 2020. My name's Suzanne Parsons and I'm the director of the Investigative Studies Program. Every spring semester, dedicated students work with faculty mentors to develop proposals for research projects that they plan to carry out during the summer fellowship process. They develop a literature-based proposal. They apply, and then the Honors and Investigative Studies Committee, made up of multiple faculty members, review the proposals. If chosen for this wonderful opportunity, the students actually get paid to do this work during the summer. They also enroll in an honors course where we teach them important research skills and life skills. And if it wasn't COVID, we would be going out to dinner together and doing fun activities, which we've done in past years. This year was a little bit different, but we still made the best of it. I'm excited to introduce to you three wonderful young ladies that I had the opportunity to work with this summer. Our first presenter today will be Miss Emily Kinney, class of 2022. Emily is a double major in biochemistry and mathematics, and she's been working as part of my research group since her freshman year here at Marietta College. In my research lab, we work in collaboration with Dr. Kevin Pate's lab in order to analyze newly synthesized paraben molecules and their effects on the viability of both normal and cancerous cells, looking for toxicity effects as well as possible cancer treatment options. Emily is working with heptoparaben, and this summer she learned how to culture cells and analyze what kind of effects these parabens are having on the cells themselves. She plans to continue this work and analyze the protein signaling pathway in both an honors fellowship project and hopefully an honors thesis in the coming years. Emily was our only sophomore to propose a project this past spring, and we're really proud of her work. Take it away, Emily.
Over the summer, I participated in the Investigative Studies Program. I worked in a lab and experimented to see how heptoparaben affected melanoma cells. I've been working in Dr. Parsons' research lab since I was a freshman, and I was excited to spend more time on my project and learn more lab techniques. Before I go further into my project, here is some background information for my project. The purpose of my project was to test heptoparaben's effects on the viability of melanoma cells and ultimately determine an IC50 value or find what concentration of the paraben kills half of the cells over what time period. The variable for this project was heptoparaben, a type of paraben. Parabens are frequently used in cosmetics and pharmaceutical products as preservatives. Parabens have a bad reputation because they have been linked to cancers as they mimic the structure of hormones. Some common parabens are shown on one of the slides. Notice how they all have a similar structure. They are para-hydroxybenzoates. The paraben I will be using is heptoparaben, made in Dr. Pate's lab, which will be pictured later. This variable, the heptoparaben, will be tested on melanoma cells. It should be noted that the article deals with many types of cells, not just melanoma cells. Melanoma is a type of cancer that develops in skin cells, but it can metastasize in other parts of the body. Melanoma cells can develop the ability to metastasize soon after they become cancer cells. This ability to metastasize makes it more lethal than other skin cancers. Because melanoma cells have a large number of mutations, they are resistant to treatments. Parabens are damaging to cells, which is why my project is using heptoparaben to kill melanoma cells. There are two main reasons for melanoma cells' ability to metastasize so easily. The first is the cell can undergo changes in its shape in order to adjust to the environment. The second component is the cell's ability to control the extracellular matrix using protease activities or breaking down proteins and peptides. Now that I have given some background information, I will tell you about the project itself. Because parabens have been shown to be toxic to different types of cells, my project investigates whether this paraben could be toxic to melanoma cells and therefore eliminate cancer. For this project, I did solubility tests, maintained a cell line, and did clonogenic assays. My first objective was to find what heptoparaben is soluble in. It was found to be soluble in methanol and ethanol. The paraben is a solid at room temperature, so it must be dissolved in a liquid solvent in order to affect the cell's viability. To test for solubility, the paraben is measured out in the amount that corresponds with the highest concentration of paraben being tested. Then the solvent being tested is put in with the paraben and vortexed. If any paraben is still seen, then it is not soluble. The solvent must be tested to see if it kills the cells. If it only kills a small amount of cells, then it can still be used, but the amount of cells that the solvent kills must be taken into account in the data. The next objective was to see how the paraben affects the cells. The cells were treated with various concentrations of the paraben mixed with the solution to determine how long it takes the paraben to affect the cells. The initial concentrations were 0.45 millimolar, 0.55 millimolar, and 0.65 millimolar. These concentrations changed as data was collected. This step involved putting the paraben on the cells and checking on them at regular time intervals. An important part of this step is maintaining a cell culture line. The cell line used was M624. The media used was made of Dobecco's modified eagle medium, 10% fetal bovine serum, and 1% penicillin streptomycin antibiotic solution. A culture of multiple plates of cells were kept by first thawing cells from cryofreeze. As the cells grow, they were cleaved from cell culture dishes using trypsin and transferred as needed. Aseptic technique was used to transfer the cells between dishes so they had more room to grow. Aseptic technique means using procedures that help to keep the cells from getting contaminated with bacteria. Then a cell viability assay, also called a clonogenic assay, which measures how many cells were still alive, was performed using crystal violet dye. The cell assay plate reader measures how many cells are on the plates by using absorbance of light at a wavelength of 550 nanometers for the purple dye. Each live cell was stained with the crystal violet dye, and the plate reader used absorbance to measure how many live cells were present. The cells that were killed by the paraben were not stuck to the well or died. They washed off when the media and the paraben were taken off before the crystal violet was put in the wells. The data gathered from the cell assay plate reader was used to determine the IC50 value. I did encounter some problems over the summer. The cells got contaminated multiple times, so they had to be regrown. This set me back some. I was also just learning how to do clonogenic assays, so my results were not reproducible, and I struggled to find the balance between the amount of time to leave the parabens on the cells for and the amount to put on the cells, because the paraben was fairly toxic to the cells and killed them quickly. I plan to keep working on this project and achieve my goal of finding the IC50 value. After completing the toxicity test, I will continue this project in order to determine the way that heptoparaben causes the cells to die. It could be apoptosis, controlled cell death, or necrosis, which is bad for the body as it is uncontrolled. Apoptosis is a natural part of cellular processes, so it does not affect the organism as necrosis does. During the summer, I also learned how to do a Western blot. In this technique, the proteins from the cell are separated by size in order to calculate what proteins were in the cell. A way to test how the cells die is by analyzing the protein content. If the cells undergo apoptosis, then they will release a protein that would otherwise not be able to be detected in the cell. The Western blot procedure has many steps, so I have not yet mastered it. I look forward to continuing this project over the next 2 years. Parabens are damaging the cells. The toxicity test that I am currently doing is a way to see how damaging heptoparaben is to melanoma cells. But heptoparaben does not just affect melanoma cells. If it were put in the human body, it could kill normal cells or mutate them. The Investigative Studies Program made me more confident in my abilities in the lab and helped me to see that I want to do research in the future once I graduate from college.
I'm Dr. Michael Morgan, Associate Professor of Political Science, and I'm here to introduce Katie Kitchen, class of 2021, who's not only a McCoy Scholar, but also majoring in both political science and English. During this past summer, I was helping Katie, as her mentor, to develop a project dealing with rhetoric used by the media and perceptions of presidential performance. And this was interesting in terms of its academic value, as she found some interesting results, which you'll see shortly, but also very timely in a heated political environment that we're in right now. So without further ado, here's Katie Kitchen.
I did my summer research on language impacting presidential approval. This kind of came out of being in an English class where we were discussing whether objectivity really exists. And then in my political science classes, we were talking about fact versus fiction and how the current political climate has sort of a war on media aspect and a fake news rhetoric coming from elected officials of all levels. Some necessary things to know based on background of literature for presidential approval right now. There are 3 theories that are overwhelming in the majority of divided government, economy, and saliency. So for divided government, this is essentially just diffusion of responsibility. For instance, if the president's party does not align with the party in the Senate or the House, that would be divided government, and voters would be more likely to blame partisanship than the president in terms of mishaps during the term. Whereas if it's unified government, so for instance, the Republican Party controls both the House, Senate, and the presidency, then the voters are more likely to hold the president accountable. So that would be impacting approval ratings. In terms of economy, it's as intuitive as it sounds, just in terms of if there's higher unemployment, lower presidential approval rating. Very basic, just in terms of financial stability for the individual levels. In terms of saliency, this works with literature on media priming and media framing. So how frequently something comes up in the news influences political attitudes and actions of the public. And this is very much just a overwhelming majority of a concept and not looking at the language specifically. But that theory is actually where I moved into mine. And so I worked with the research question over the summer of how the level of emotional language in national news publications influences the American public's approval rating of the US president. And so I moved from that into working theories, both with English and political science backgrounds, as I'm a double major in that, and looked at how word choices frame concepts. So you can use a different framing or a different lens and use different words to create a different reaction in your reader. And this is unintentional or intentional, depending on whether it's manipulative. But just stepping back from that, it's just you get a different reaction based on the languages that you're using. So I stepped from that and looked specifically at emotional language. So I looked at 4, which would be positive, negative, anxiety, and anger. And those are just the key concepts that the program that I was using had. And so you're able to quantify something that's generally very qualitative to get statistical results. So I went from that and looked at how immediate the reaction would be to set up my study. And priming and framing generally happens very quickly. So you'll read something and have a very quick change in attitude or influence. And this adds to current literature as it's a digital focus. I use the archives from the New York Times. And then it's rhetorical content analysis. So as opposed to the theory on saliency, where it's just the number of articles, it's actually looking at the specific language and words that are being used. From that, I had 4 hypotheses, essentially just showing directionality of language. So I hypothesized that an increase in positive emotional language would then, in turn, increase presidential approval rating. The reverse of that being an increase in negative emotional language would then decline presidential approval ratings. And then for anxiety, I also hypothesized a positive relationship, meaning as anxiety language increases, so would presidential approval. And I hypothesized that as angry language increased, approval would decline. And then I'm able to test that direction and see if there is a correlation between the two using statistical software. The methods that I kind of went through was to choose New York Times, collect articles from that, and then I inputted them into linguistic software. It's a word count software called Luke. Then I condensed all my variables and was able to put all of the output from that software into Stata, which is the statistical analysis. To gather the articles from the New York Times database, I did a keyword search for the U.S. president and the sitting president's last name. And then on the 1st, 7th, 14th, 21st, and 28th day of every month from 2001 to 2018, I grabbed the first article which came up with those keywords so that it was very much an objective and not a subjective type of gathering data. And I made a sheet of all that and then got the output from Luke. I have a chart here that really demonstrates what a Luke output looks like just on an overview level. And so you can see where the X's are. That's where Luke coded a dictionary to show that those words are from those categories. So this is an article on the left-hand side. There are a bunch of words, and that's actually an article that I used in my study from January 2012. And you can see that the word fights is a negative emotion, and an angry language. And these have X's beside them, which Luke then in turn puts into a numerical data in percent form. And you can see that strongest is the opposite of that, and it has a positive emotion, X. And so that's how it brings out every article from 2001 to 2018 and is able to give me a quantitative value for a qualitative language. This chart shows all 4 types of emotional language and just their trends that Luke was able to output from 2001 to 2018. You can't really see a visual trend, which is the really cool part, is because when you take this and then you put it into Stata, you can get results that you can't see with your eye, just guessing at it. So these are all 4, and you can see that there is high amounts of movement through them. The following graph is just the presidential approval from 2001 to 2018. And you can again see the large spikes and declines happening as approval is something that changes rapidly and was actually collected daily from the Presidency Project in Gallup from 2001 to 2018. And so the vertical lines show the end of President Bush's term and the end of President Obama's term. That way you can just get an idea of what it looks like in those time spans. Moving from that, after inputting them into Stata, this is the chart that you'll see, and it's actually like the most important part of my study is this is the results. You'll look on the left-hand side, and there are a bunch of words down there. The first 4 are my independent variables, so it's the 4 types of emotional language that I used. Everything following are called control variables, and these are essentially necessary to ensure that my study isn't overestimating the value of what I'm adding to current literature, and it just uses unemployment, as I said, economy, and then saliency. I used the number of news articles that included the sitting president's last name as opposed to anything actually dealing with the language. For divided government, I used House and Senate seats that align with the president's party. Arguably the most important control variable that I use is the approval lag, which is my dependent variable, but set off by a month. And this is intuitive because approval from one month is going to influence the approval of the next month. They are very tied concepts, and if I would fail to include this, it would invalidate my results as it would highly overestimate the level of statistical significance that my variables had. Moving just into what I found throughout my study, the important parts are the yellow highlighted column and the red highlighted column, and this is the T and P values. For T to be statistically significant, you want the absolute value of that column to be above 2. But for P, you want it to be less than 0.05. And as you can see, with my 4 independent variables, all 4 are statistically significant. For positive emotion, it's in the direction that I hypothesized. It's a positive coefficient and it's significant. So that means as positive language is increasing in the news, then presidential approval is also increasing. For negative, it is also how I hypothesized. As negative increases, presidential approval decreases. Basic, but it's now statistically proven. And then for anxiety, it is how I hypothesized that as anxious language increased, presidential approval also increased. For angry language, actually, it's the opposite direction that I hypothesized, which might not make sense, but after further looking into literature, there is an explanation. So as angry language increased, presidential approval also increased, which can be explained by the fact of the rally around the flag effect. And this is what I had hypothesized anxiety positively for, but I hadn't known that anger correlated with that concept so much. And it's just like in a time of peril or anxiety or anger, for instance, 9/11, the voters and the public tend to rally around a central figure, which is often the president. So those 2 emotions being so tied to that effect, that's why they have a positive influence on the presidential approval rating. Just in like a broader discussion of that, my study proves that news is riddled with emotion. So objectivity really isn't existing in that atmosphere. And then legitimacy of the president, that's based on public approval ratings. So like authority and legitimacy to make decisions, this as an elected official is very much set in how confident the public is in you. And then that just shows that language is significantly impacting the effectiveness of our democracy. So language changes attitudes of Americans, which changes so many things about how they approve and how they vote, which I think makes this study really prevalent in this atmosphere right now, especially with the election coming up, because increased public approval rating or decreased public approval rating from just the language that's being written right now and being read right now is going to change the attitude of the public when they go to vote at the polls, because it is such an immediate reaction. So I think that being able to inflate or undermine public approval in a sitting president or a presidential candidate is something that we should definitely take note of. In terms of future considerations, I think looking at new media sources, which would be like social media or different types of online blogs, as opposed to a traditional media source, which I use for New York Times, would be an interesting factor, as well as with presidents more frequently having accounts of their own on social media. That's allowing them to have a personal relationship with the public, and it's kind of taking out the media from the center source. So looking into the future, if that trend continues, how that impacts the media's role would be an interesting thing to look into. In terms of individual charisma, there is some literature out there on just saying this president's likable, and you can't change that based on anything that they do, and my study doesn't account for that. However, it's not always statistically significant, so maybe finding a way to control for that variable would also add to my study. And then lastly, so I did systematically gather the articles, and I used a very objective word count software. However, I can't always ensure that the angry language that's in an article is always directed at the president without going through and reading that, which would then make it a very subjective process. So finding a way to go through that and take out that possible conflict would really clarify some results as well. And then just some final acknowledgements of the Investigative Studies Program for allowing me to do my research over the summer. Dr. Morgan, the political science professor who was my advisor throughout this, and then just the political science department for buying the Luke software and pushing me to do this. I definitely appreciated the opportunity to be doing this, and I learned a lot about the software that's necessary in my field, as well as just how to take a theory that I just came up with in class all the way through to finding statistically significant results that adds to current literature in my field.
Our last presenter today is Miss April Davis. April's a member of the 2021 class and a biochemistry major. She's also a volleyball superstar and currently in the process of applying to medical school. Go April. April's project was different from really any student project that I've done here at Marietta College. April decided to work on an experiment that we started in spring semester in one of my courses. In Chem 426 Advanced Biochemistry Lab, we performed an experiment that didn't go so well, to say the least. April was determined to figure out what we did wrong and how to make this experiment a success. It was really cool to work with April this summer because I got to work alongside her in the lab and learn new techniques that I had never performed before, which was a really cool experience as a research faculty mentor at Marietta College. Take it away, April.
My research project was focused on the purification of the enzyme alcohol dehydrogenase from yeast cells. It is important to note that baker's yeast is used in plenty of foods, including cakes, sourdough breads, and alcoholic beverages. Alcohol dehydrogenase is an enzyme that is responsible for speeding up the rate of a chemical reaction. More importantly, it catalyzes an oxidation-reduction reaction in nature. It reduces NAD+ to NADH, which is what I measured to determine the rate of the reaction. You might be wondering why purification of enzymes is important. There are three main reasons why it is significant. First, it allows for the specific study of the enzyme by allowing the structure to be determined. It also helps to figure out other important biochemical traits about the enzyme. Second, purification is used in medical and industrial purposes. More specifically, ADH is used to treat drug addictions. It is also beneficial in treating respiratory infections. Another common use is bovine insulin has been used to treat diabetes before scientists discovered how to synthesize it. The third reason purification is important is to study enzyme kinetics. This allows determination of the reaction rate of the enzyme, which was evaluated in my research project. It also allows researchers to understand the enzyme's role in metabolism. My actual research project was focused on determining how to purify the ADH enzymes from yeast cells while maintaining enzyme activity. There were numerous procedural steps that I performed in order to purify the enzyme. First, the conditions in which I grew the yeast were manipulated in order to determine the ideal temperature, pH, time, and buffer used. The other important factor was which part of the broth I took my sample from once the yeast cells were grown. I then performed an immobilization process which was designed to protect the enzyme from other changes made in the experiment. The second step was homogenization, which is breaking apart the cells to release the enzymes from inside. Salt fractionation was the next procedure that I performed, including both salting in and salting out, which is essentially increasing the level of salt concentration of the solution in order to cause the proteins to precipitate and fall out of the solution so that they can be collected. The next procedure was dialysis, which was fairly straightforward with not a lot of options for manipulation, but it includes separation of the ideal proteins from other cellular debris by using dialysis tubing. The last procedure was column chromatography. Again, there was not much room for manipulation, however, I performed the procedure multiple times in order to perfect it. It is valuable to understand how I measured activity in order to comprehend how I evaluated the data to attain the rate of the enzyme. When measuring absorbance of each sample, the reduction of NAD+ to NADH is significant because NADH absorbs light at 340 nanometers. Therefore, the UV-Vis spectrophotometer is used, which shines light through the sample and determines the difference between the original amount of light and the light that comes out on the other side of the sample. The difference is the absorbance of light by the sample, which theoretically should increase as the concentration of NADH increases in the sample. Therefore, this procedure allowed me to quantify the rate of the reaction performed by the enzyme alcohol dehydrogenase. As previously mentioned, the growth of the yeast was analyzed to determine which conditions result in the highest absorbance values. After growth, the yeast solution was left to settle in order to determine which layer would have the highest absorbance. My results showed that the bottom layer of the liquid solution resulted in the best absorbance value. Therefore, that is where I chose to take my yeast samples for the rest of the experiment. The next procedure that I performed was immobilization, which is attaching the enzyme to an insoluble material in order to protect it. It actually changes the appearance of the yeast cells. By doing this, it increased the resistance to changes in the environment such as temperature, pH, and salt concentration. My results determined that the immobilization procedure was effective at increasing the absorbance of the yeast samples by over 3 times as much activity. Homogenization was the next procedural step that was analyzed. This included using a mortar and pestle in order to grind up the cells. I compared frozen cell samples to room temperature samples to determine which results in the highest absorbance values. My results determined that frozen ADH samples led to higher absorbance values. The next procedure I analyzed was salt fractionation, which is increasing the concentration of salt in the solution in order to eventually get the desired proteins to precipitate and fall out of the solution. There are 2 steps, salting in and salting out, and by the end, the ADH enzymes should be in a pellet at the bottom of the beaker. My results showed that salting in at 20% and salting out at 40% worked best when using potassium phosphate as the salt. Dialysis was the next procedure performed, in which I used standard weight dialysis membrane in order to separate the molecules by size. The results did show that the separation technique was successful because the ADH enzymes were found in the tubing instead of in the solution. Column chromatography was the final process performed, in which the molecules were separated based on charge because of stationary and mobile phase within the column. The results showed a slight increase in the absorbance of the sample after it passed through the column. Another aspect of the experiment that was necessary to study was the storage of the ADH enzymes overnight, because the entire procedure cannot be performed entirely in one day. Moreover, in order to determine the decay in enzymatic activity, the sample was stored in the freezer overnight and the absorbance was measured each morning using the same technique. The results showed that either the constant freezing temperatures or the process of thawing and dethawing denatured the ADH proteins, because after 1 week, the sample was barely able to absorb any light. These are all of the results that I obtained during my summer research. I hope that I can continue this research throughout my senior year, as well as conduct a similar experiment for my honors thesis. As far as experiences, I learned so many valuable lessons throughout doing this research project, and I would really encourage all undergraduate students to take advantage of this great opportunity. It taught me persistence as failure continued to happen. I had to figure out a way to work through them in order to reach success. It also allowed me to figure out more about my passions and what interests me in the biochemistry field. This project also helped me to prepare for medical school because I improved my research skills as well as my planning and organization. The Investigative Studies Program is one of the best opportunities to prepare you for your career after Marietta College. I would like to acknowledge Dr. Parsons for helping me through this entire research project, as well as getting me all of the resources necessary to perform the experiments. I would also like to thank the Marietta College Department of Chemistry and Biochemistry for all of the resources, lab space, and equipment that they provided. A big thank you also goes to the Marietta College Investigative Studies Program for giving me the opportunity to perform this research project.
That wraps it up for Fall Symposium 2020. Thank you for joining us. I hope that you found these presentations interesting and inspiring. As you can see, undergraduate research is thriving at Marietta College. If you're a current undergraduate student, please reach out to your faculty members, your faculty mentors, and see what kind of creative work or research work you can get started in today. The Investigative Studies Program is here to support you. Thank you so much for joining us, and I hope you have a great rest of the fall semester.